speak after group photo. It was nice of you guys. So let's start the program. And we miss one speaker because her health is not well. So we wish next time she will come. But the other also positive side, we covered 15 minutes, so don't worry. Okay, let me invite our first speaker, Professor Dominik, to share his research and ideas. Prof Dominik, please.
Thank you so much, Siad, for having me here for 2 weeks and for giving me the opportunity to present my research on this conference. I know we are short in time, so I think I will skip official presentation. You already know me, right? And let's go to science. Actually, this is my department, which is pretty big. We call it faculty because we have about 60 professors on the department. And yeah, and I think maybe it's different than here. We have like 200 first-year students, and only 80 of them survive. So that was the very brief summary of the faculty, and now let's go back to science. So I meant this talk to be quite general, and typically in our review papers, where we review what we are doing, and also some review presentations, we are showing a graph like this, and this graph presents time scales and dimension space of biology or any event that is biologically relevant or relevant to life sciences. So here you have quantum effects like bond formation, bond breaking, reaction, enzyme catalysis. This happens in femto to picoseconds, and it's on a very, very short scale in terms of distance. And later you have movements of atoms, movements of chemical groups, movement of secondary structure elements of proteins. Later you have, of course you have diffusion, but later you have protein folding, which can take up to a millisecond even. And later you have global motions of big moieties, like ribosomes, and like protein synthesis and so on. And finally you have very, very time-consuming events, events that take really lots of time, like protein aggregation, which now is in Europe is a problem for our societies are getting older, so we have the problem with diseases that are related to protein aggregation. So we also want to study these events. Unfortunately, these events take even years in life, in real time. So it's from theoretical, experimental point of view, there is no single experiment that would cover all of these phenomena, and you need many experimental techniques to study them. It's what's called today morning. It's a computational conference. So let me focus on computational approaches to these phenomena. So also, you cannot study all of these events with a single method, computational method. You need many of them. You have different approaches to do quantum calculations, which it's not my field. So let's keep it for this talk. Then you go to all atom calculations, where every atom is like a hard sphere. The spheres, they kick each other. There are bonds, the bonds vibrate and so on. You have forces, you have like real physics. I hope you are not scared of it. So this is this area. And we use these methods, we don't develop them. Mostly we are focusing on these coarse-grained approaches. I will tell you about this more later. And we also have mesoscale. When you don't have atoms, you have just continuous space with some properties. And we are not dealing this too. We are just focusing on these two middle boxes. And with these methods, you can study protein dynamics and protein interactions and protein folding. So this is what we do. We are mostly interested in protein structure and protein dynamics. And we try to go to as big proteins as possible, as big protein complexes as possible. So this is what we do basically. And there are two modeling worlds, or two approaches to protein modeling. One of it is it's physics, So maybe you had some physics course. For sure you had then some formulas on energy. In high school you maybe had like potential energy, kinetic energy. Later you learn about electrostatic energy, about weak interactions, London forces, van der Waals and so on. So this is like energy, you find these formulas in the textbook. You have Newton laws, so since you have energy, you have force. When you have force, you have Newton law, you have acceleration, you have velocity. You can model how the whole system evolves, how it behaves. And this is really nice, it's fully justified. So guys from this world, the world of physics, they claim that this approach is fully justified. If there are any other simplifications or approximations, they are clearly stated in the formulas, all is clear, you can describe everything. The problem is that you can't solve it. Yes? The formulas for a big system are too complicated to be even numerically solved. And even if you have really big computer, you can hardly simulate a protein folding actually. It's not that easy. And another approach is by biology or by evolution. It's totally different approach. So there are no general rules. You just learn by example. You guess by similarity, by homology. So if I have two proteins, and for one of them I know the structure, I know the function, and if the two are homologous, this means that they evolved for common ancestor, and I believe that if the evolutionary distance is not too far, if they are not too far from each other, then they still have the same function. So when I teach bioinformatics, I always give this example to students that let's say that you are somewhere in the medieval times and suddenly you saw a car. Of course you don't know what's a car, yes? There are no cars in medieval times. So you start scrapping it and you see the engine. You don't know what's the engine, of course. You don't know what's the function of it, yes? You see a driving wheel. You don't know what's a driving wheel, yes? So everything is totally new for you. But when you saw 10,000 cars, different cars, when you scrap them, you will see that you have 10,000 engines and you will start seeing the difference. Maybe you have 200 diesel engines and 800 gasoline engines, yes? So in terms of bioinformatics, you have proteins and you cannot say what they do. But you see that the sequences are similar. So you have engine family, you have driving wheel family, you have tire family of those proteins, yes? And then you start like dividing them into subfamilies. Like a subfamily of diesel engines and subfamily of gasoline engines. So you just learn by cases you see. So the more data you collect, the wiser you are, basically. Of course, you can learn the function, and if you know the function of one protein in a family, then you can extrapolate and say that other proteins from the same family share the function. So this method works very well, and it works only if you have the data. So I also teach students that this approach is like you have the equations and you solve it. And this approach is that you actually have answers book and you cheat. But the problem is that your problem, your sequence, sometimes it's different, or in many cases it's a bit different than the cases you have solved. So you have to adjust the answer, yes? But this is just a matter of small adjustments. If you have one protein from a family, and you know the structure for it, and you have a sequence from the same family, typically you adjust the sequence and to build a model, a structure that is similar to the protein you already know. So I would say it's like a bit cheating, yes? Guys from the physics world, they say that these guys don't understand the rules, these guys don't understand why this protein has this particular function and not the other. Because these guys, they don't talk about forces, don't talk about interactions, they just learn by example. Nevertheless, this approach is way much more successful. So if you have the data, use it. So I have two examples to illustrate these two approaches. One is really simple. This will be a Monte Carlo simulation of argon gas. So as I told you previously, you have some energy equations, you have some law of motion. In this case, we don't use Newton law, we use stochastic sampling, but it's more or less the same. you have some temperature, some density, and energy. This is energy equation. So any two argon atoms that are in distance r to one each other, you can calculate the energy of the interaction according to the formula. You have a plot, this energy, how the potential looks like. So may we have the first movie, please. On the movie you will see, yes, this one, you will see a simulation of argon liquid. This is just an argon droplet. From time to time you will see the order. Like now you see that the balls are like in a plane. Yes, they just queue up in lines. So they like form a crystal. Sometimes you can see a crystal lattice, like now. Sometimes you can see disordered droplet, so then it's a liquid. So this is a phase transition between a liquid argon and a crystal argon. In the simulation, you just have one droplet of argon. You see 4, actually this is 9, we use 9 because there is a central, so 3, actually no, we use 27, it's 3 by 3 by 3. So the middle one must have neighbors. Yes, so this is a simulation trick because otherwise we would have too small system. So we copy the system 27 times so that we observe only the middle one, and the middle one is recorded here on this movie. You have 4 just to, this is actually the movie I use for teaching. The middle one, let's say it's this one, it has neighbors, and this you can see sometimes evaporation. So even in cold temperature, argon can evaporate from crystal or from droplet and can move away, and sometimes it actually gets back to the droplet. So yeah, okay, not this one, I have a presentation. So as you, no, yeah, the PDF please. So as you saw here, in physics-based simulation, atoms actually move. You solve some formulas, and you have a trajectory, we call trajectory. We have coordinates of every atom, every time, every couple of, let's say, femtoseconds or picoseconds, you have a state of the system. Bioinformatics, it's totally different. Yes, in bioinformatics you have lots of data. I think some of you already saw this is a multiple sequence alignment of a protein family. It's just one sequence, it's the same protein from different organisms. So these are homologs, they are aligned with a program. So every one column actually corresponds to a single position in the protein. You can imagine that, let's say 3 million years ago or 10 million years ago, there was just single protein, and the protein has position number, let's say 58, and in this position 58 there was, let's say, lysine, and during evolution this position was changing. Of course there were also deletions and insertions, so the position is no longer 82, sometimes it's 110, sometimes it's different position, but in the alignment we introduce gaps, like here. So the same position is all the way from top to bottom in the alignment. So it's the very same position that happened millions of years ago in the very first protein from this family. And this is colored by chemical properties. So I think aromatics and non-polar residues are blue, and charged residues are in pink, and polar are in green, and glycine is orange. So you can see that chemical property is conserved. This is really important. So for example here the letters are different in the columns, but it's blue all the way from top to the bottom. Yes, because this part of the protein may be, for example, inside the protein, and it must be, it cannot be polar. Yes, it must be aliphatic or aromatic, because it forms hydrophobic core of the protein. So it's all the blue. So evolution imposes a constraint, and during evolution amino acid changes to different amino acids, but it's chemically it must be apolar. So this is, we can learn many things from this multiple sequence alignment. Based on the multiple sequence alignment, we can make a phylogenetic tree. You will hear about this later today. But okay, so what can you do with bioinformatics? So that's also another example that I use for teaching. You have a bunch of species. Since I came to Africa, I think African species are overpopulated here on this slide. So now this is a case study from my students. Which of well-known species mammals that live on land is the closest relative to dolphin? We all know that dolphin is actually a mammal who lives in ocean. Of course, he has many relatives on land. I mean, related species. Which is the closest? I'm afraid I'm running out of time, so I don't let you guessing. I tell you that my students typically guess that phoca is the closest one. But unfortunately, it's not the case. The closest is hippopotamus. And you can learn this by doing a phylogenetic tree and just checking the difference between genomes. So the closest the genome, most likely that this is the closest relative, yes? Because the genes didn't have enough time to evolve apart. So this is what you can learn from biology. You can also learn, I've told you about this, you can learn a structure of a protein for which you have only sequence. You have an input sequence, you find a structure that is homologous to your sequence, and for this structure you must have, for this protein you must have structure, and you call this a template. And then you align a template with your input sequence. Basically you make something like this. One of these sequences is your template, one of the other is a target or query that you model, and then you build your model structure, you refine it, and you assess it. If it's okay, then you are done. If it's not okay, then you repeat the whole procedure. This is called comparative modeling, and this is the best way to get a structure of a protein on a computer, providing that you have a template. And this is really strong assumption. I mean, we have like 10 to 8 power of sequences already discovered, and only about 130,000 of structures. Yes, compare the numbers, yes? 100,000 structures and 10 to 8 power of sequences. So, of course, there are many sequences for which you cannot do comparative modeling procedure. And there's lots of software packages that can do this. Some of you might use Modeler. Rosetta also can do this. And we use another tool called CAPS. I hope I will have time to cover this too. Yeah, so this is Rosetta community. Rosetta is also a software we develop. I know that there is a dress competition, and I should dress to kill, but this is my uniform. I have this Rosetta t-shirt today. Most of these guys, they also have Rosetta t-shirts. This is from Rosetta conference. So yes, we do develop Rosetta software for comparative modeling, but actually comparative modeling is not the main application of the software. Main application is protein design, as it was spoken today. Bioinformatics is really big business now, especially in protein design. You can design your own enzyme that catalyzes your reaction and produces the product you want. So come back to this. So if you want to make an all-atom model of your protein, then do comparative modeling. You cannot probably make this with all-atom simulation because it's too slow. So approach of my group, I focus like 50% of the time of my research time is coarse-grained approach. I will show you one example. So this is all-atom protein is simulated by molecular dynamics. Every atom is a sphere, basically. So to the other extreme, we have a surpass model where one residue is one sphere. So there are some forces, some energy defined that rules the system. It's much simplified. Let me emphasize, each residue is a sphere, and here each residue has like 20 spheres. Yes, because amino acid residue has between 10 to like 20 something atoms, if you include hydrogens. So this is like 20 times less atoms, so it's like 400 times faster. It scales with the second power. So this is an example. This is 2GB1 protein all-atom representation, and this is just in surpass representation. We have average interactions, so may I have the second movie. On the second movie, you will see dynamics of the protein in surpass representation. And on this movie, you will see how secondary structure elements are formed. And this movie, calculation for this movie took like 15 minutes on a laptop. So this is really, really fast. This is like 1,000 times faster than all-atom simulation. This is really, really fast. Of course, biologists are not happy with these spheres. Of course, they don't know what to do. But we have software to recover, to recapitulate all-atom representation, to rebuild whole atom structure from these spheres. Because protein, when you fold the protein, you have to pack everything nice. So the problem is like you have a piece of wire or a piece of cable, and you want to do like this, and put it into your pocket, and believe that the structure is like the native one. Of course, yes? So what we are doing, we're just sampling all the conformations, looking for the native one. Can we go further? I mean, the movie is quite long. But this just gives you the feeling what we are doing in simulations. But also we do data processing and bioinformatics. I developed BioShell package that does sequence alignment, structure alignment, structure comparison, hierarchical clustering, lots of analysis. It processes lots of different file formats. I'm afraid I have no time to dig into this. I have one example of what BioShell can do. This is one of hundreds of examples. Let's say you have a protein that has been crystallized with EP molecule. This is just a buffer, so this is not a real ligand. This just comes from crystallographic buffer. And you have templates, and in some of these templates you have real and in some of these templates you see the real ligands. So BioShell is able to compare all these structures, and after the analysis is huge numbers. So this is what I could stress. BioShell is good in mass processing of huge number of proteins. You discovered that this buffer ligand actually substitutes real ligands in other proteins. Okay, so I expected I will be late, so I'm just going to conclusions. First of all, I want to tell you that databases of sequences are growing way much faster than the speed of computers. Of course, computers are getting faster, but data is coming faster than we can process it, so we need smarter algorithms. Also, I show you two ways, but these guys typically don't talk to each other. This is bad. I think the best approach is to join physics-based approach with bioinformatics approach. This gives you the best result. And so far, Unix works of style dominates, but we try in my group to do a graphical interface to our software. And of course, don't forget about BioShell package. Please ask me about applications. I have a nice gallery of applications that BioShell can do. You can download the software from, and there is a documentation here. And I think that's it.
Thank you so much, Prof Dominik. We can have brief 2 or 3 questions from audience. If anybody would like to ask, please raise your hands.
Dominik, your Rosetta software, does it also cater for nucleic acid studies? I see that you've been talking so much about protein structures. How about nucleic acids? Can you
so the question was if Rosetta can model nucleic acid. So, in general, yes, but it started with proteins. Applications to nucleic acids are newer and not that well developed. What you can model protein-DNA interaction. You can also design de novo RNA structures, small RNA structures. And probably you can predict RNA structures too. DNA is, people think that DNA is boring because it has always the same structure, right? I mean, there are like 3 or 4 forms, and that's it. The most interesting part is protein interacting with DNA. Proteins interacting with RNAs are the most difficult because you first have to get RNA structures right, which is tough, and then to be able to dock it to PDB, to protein, which is also tough. But protein to DNA is, I would say, is now standard for Rosetta.
Have you thought about maybe optimizing the whole nucleic acid protein interaction and making it automated online for instance? Just like with homology modeling, there are so many softwares that you can use to model a protein, but there are also softwares like Swissbdb where you can get an already modeled structure online. You don't have to do that by yourself. Have you thought about making a tool that is automated?
Yeah, so the question was whether we can automate. And the general answer is no. The specific answer is yes. For a very specific case, you can make an automated tool. For a very general case, as I said, the Unix style, terminal style work is really popular in this field because it gives you the highest flexibility ever. You can play with thousands of parameters. Of course, you don't want to play with thousands of parameters. But if a tool is to be used for many different applications, that you need to play with these 1,000 parameters. So, what, for example, can be possible, you may have like a protein that binds, like a transcriptor factors that bind to DNA. And it's possible to make a web-based tool only for this. Just specific protein or specific family of proteins and specific selection of DNA sequences. Yes, then it's possible to make it online. Cool.
We can take one more question. Could you please use mic when you are asking question so he can hear, please? Thank you.
Thank you. I just want to know if from your platform one can be in a position of simulating histone tails, as they are important in controlling or regulating gene expression.
So, histone tails or the whole histone proteins?
The tails, because to a great extent, you find a nice movement of these tails.
Yeah, so as for tails of proteins, of any proteins, this also includes histones. The problem is that bioinformatics cannot help you. Yes, because these fragments are unstructured, and even if you have a template, this doesn't help you. And you're interested in dynamics, right? So, in principle, yes, you can use like molecular dynamics for this, but this needs high CPU power. Because especially with DNA, if you need dynamics or something in physics-based approach, then you have to load your system with water, ions. Like on this movie I showed you, there was no water. Water is just averaged, because it's not that important. And no ions, no buffer. It's not that important in protein folding. But in DNA, counter ions, with DNA is highly charged. So you need counter ions, you need water. It's complicated, but it's doable. Yes.
If you are anybody interested, please interact with Prof Dominik. Let's give a big hand for the excellent talk. Without delay, let me call next speaker.